Hello everyone, I'm Sheena and welcome to my channel Comprehend Biotech. In today's video, we are going to learn about an experiment done by Frederick Griffith. Why is it important? Because Frederick Griffith's experiment on transformation actually laid the foundation of how scientists discovered that DNA is the genetic material. So this video is a kind of back to basics video where you will understand the basic concepts of biotechnology. So this particular series is named as Back to Basics. So you might be a high schooler, you might be just starting your undergraduate study in biotechnology field, or just anyone in general. This video is just for you to understand a very basic experiment describing how Frederick Griffith found out the transforming principle and in turn took the first step in finding out that DNA is the genetic material in a cell. So let's start. So generally, when we talk about DNA being the genetic material, there are majorly three experiments that actually prove to us that yes, DNA is genetic material. So those three experiments are Griffith's experiment, Oswald, MacLeod and McCarty's experiment, and Hershey and Chase's experiment. Hershey and Chase's experiment was something that sealed the deal, and we finally got the proof that yes, DNA is the genetic material. So in today's discussion, first we'll be starting with actually what do you mean by a genetic material? What are the characteristics of a genetic material? And of course, what is Griffith's experiment? So what is genetic material? So this is a question to you. Ever wonder why you look like your mom or dad? Or why do you have certain characteristics similar to them? Now, the most fundamental property of all living organisms is to reproduce, right? So all organisms inherit their genetic information, specifying their structure and function from their parents. So likewise, all cells arise from pre-existing cells. So the genetic material must be replicated and passed from the parent to progeny cell at each cell division. Okay, so basically something is being transferred from your parent to you, and that is why you inherit their characteristics. And that is why sometimes you hear, hey, you're the carbon copy of your mom, or you're just an exact copy of your dad. Or even if you might not look like them, you do possess certain habits, certain characters, which, are typical of your mom or your dad. Something like that, right? So basically, whenever a cell divides, the genetic material is passed on from that cell to the next progeny or the offspring cell. And that is how all the information that is present in the parent cell is passed on to the progeny cell. So what is genetic material? Any material of plant, animal, microbial, or other origin that carries genetic information and that passes it from one generation to the next is called the genetic material. Now, the genetic material must be capable of carrying large amounts of information, replicating faithfully, and translating its coding instructions into phenotypes. So these are the three requirements of any material being the genetic material. First of all, it should be able to carry a lot of information, right? From everything. Just look at the example of a human being. All the information starting from how you should look and what should be your eye color, what should be your hair color, how should be your skin texture, how should your digestive system be, how should your organs function. Everything, all that information should be present inside the genetic material, right? The second property is that it should be able to replicate faithfully. So if you look at your body itself, your body is made up of different types of cells, right? You look at your skin cell, it is a type of cell. So whenever a skin cell divides, right? So just imagine you were a baby once. From a baby, you have grown up to such a huge human being, right? So your cells have been continuously dividing. And the best part is that each cell is carrying the same genetic material. Today, we know that the genetic material is DNA, but back then, we didn't know what it was. And mostly people thought that the genetic material was actually protein. Okay, we'll discuss why that was the general notion. So that's what is being said, that the genetic material should be able to replicate faithfully. And the third thing is it should be able to translate its coding instructions into phenotypes. So basically, whatever information is there in the genes or in the genetic material, it should be able to be expressed or translated into what we need for our body. If by now you might be aware of the central dogma of molecular biology. DNA is transcribed to messenger RNA or mRNA is translated into proteins. So that a process where you get a protein from a gene. That is called gene expression. So a genetic material must be capable of being translated into a particular information. Finally result in a particular phenotype. Phenotype is the physical features that we see on us, right? Physically, what is the effect of a gene? So which is the genetic or hereditary material in a cell? So for almost 100 years, scientists had known plenty about proteins, okay? They had known proteins were of different shapes, sizes, functions, and okay. So for this reasons, many scientists believe that proteins were the hereditary material. Another reason was, nearly 50% of your cell is composed of proteins. So people naturally thought that, if so much of information has to be passed on, it should be carried by protein because the protein content is really high. So by then, the discovery of nucleic acid was already done when, people were trying to find out what is the genetic material. Nucleic acid was already discovered by Measure, okay? They had even found out that, nucleic acids are composed of 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. Whereas a lot of studies had already been conducted on proteins, and they knew that there are nearly 20 amino acids. So when they were comparing all these data, they had a complete notion, all the scientists and researchers, they had a preconceived notion that protein has to be the genetic material. Look at DNA, look at protein. Protein is abundant in a cell, DNA is not that much. And there are 20 different amino acids which can code for a different type of protein, but there are only 4 nitrogenous bases. What combinations could it be in that it can carry such a huge amount of genetic material? So that was the confusion. So it wasn't until 1928, when Frederick Griffith identified the process of transformation, that individuals started to question this concept. Now, Griffith demonstrated that transformation occurs, but what was the material that caused the transforming process? So here I want you to know that the experiment that Griffith conducted, he just came up with the concept of transforming principle or transformation. He didn't actually find out that DNA is the genetic material, but his experiment on Streptococcus pneumoniae was taken up by other scientists, which finally gave us the perfect idea that yes, DNA is the genetic material and not the protein. So that is why I keep telling that Griffith's experiment is kind of a stepping stone towards the discovery of DNA being the genetic material in a cell. Okay. So Frederick Griffith's discovery of bacterial transformation. So this phenomenon of transformation was first observed in 1928 by Frederick Griffith, an English physician whose special interest was the bacterium that causes pneumonia, known as Streptococcus pneumoniae, right? So he was studying this particular microorganism or this particular bacteria which causes pneumonia, and he had isolated numerous types of strains. Strains means different forms of the same bacteria, okay? So the strains that he formed, for example, he found something called the R type strain. So you might be knowing that bacteria generally grows as colonies, right? So one single bacteria, it multiplies, and over a period of time when you observe it, it appears as colonies. And how the colony appears helps us to distinguish different types of microorganisms. So Griffith had found 2 different types of strain. One was called the rough strain, one was called the smooth strain. So the rough strain was called so because it appeared that it formed some rough type colonies on the plate, right? And the S strain or the smooth strain actually formed certain smooth colonies. Okay. Now, what was the difference between them? Basic difference was that the R strain did not have a polysaccharide capsule around it. And because S strain had a polysaccharide capsule around it, it was giving a smooth appearance, or it was showing us smoother colonies. And one more difference, major difference, was that because it lacked a protective capsule, R strain was whenever it enters any body, for example, any organism, maybe humans, it is not able to infect, because the moment this bacterium enters our body, it can be killed or it can be destroyed by our immune system. Right? But S strain is not like that. S strain has a protective coating, right? Polysaccharide capsule. So it is a virulent type of strain. Okay, that means it can cause infection. So whenever a person or any organism is infected with the S strain, generally that organism will develop pneumonia and the organism generally dies. So these experiments were conducted in mice, So Griffith injected various strains of this bacteria into mice and he was studying. He was just generally interested in studying the strain about pneumonia. He didn't know that he's going to discover something so incredible, right? So what did he actually do? So as I mentioned, he was working on Streptococcus pneumonia and he had isolated different types of strains. So one of the strain was type 3S. That means it was a smooth strain, S strain, and it was type 3. He just gave it different names, okay, type 3. And then he also started working with a type 2R or rough strain. Okay, so basically he was carrying out different mutations and stuffs, and he always found something very interesting that type 2 R strain. Type 2 R strain could be mutated and be changed into type 2 S strain, that is virulent strain. Okay. And type 3 R strain could always be mutated into type 3S strain. But never had he found that, in any experiment that type 2 R strain is being mutated into type 3. The type will always remain same. Okay. 2 doesn't become 3. Uh, if it is a type 2 R strain, it will be always generally becoming or mutated to become type 2 S strain. Type 3 means it will become type 3 and never does a type change from 2 to 3. Okay, that is what he noticed. So he started working with the type 2 R strain, which is rough strain and which is a non-virulent strain. It does not cause an infection. And he chose the type 3 S strain to work with, that is the smooth strain, and which causes infection. Then what he started doing was he started injecting these various strains into the mice. And now let us see exactly what he did and what did he discover. So let us understand what Griffith's experiment on discovering the transforming principle was by looking at this little whiteboard behind me where a structure has been drawn which actually represents a mice. So please in your imaginations keep that this is a mice, okay? So there are 4 sets of experiment that Griffith actually did, and we will see each type or in each set what did he actually do, okay? So in the first set, basically he took live R strain. So if you remember, the R strain or the rough strain, it is generally a non-virulent type. It does not cause pneumonia, okay? It does not cause any kind of infection, so it is non-virulent. So he took that strain, he injected it into a healthy mice, and then he waited for the results, okay? What happens? Now Griffith was an intelligent man. Of course he knew that since it is live R strain that he injected into the mice, of course there was no pneumonia or no infection. And since there was no infection, the mice lived. A very straightforward experiment, right? The mice lived. There was no infection, so the mice lived. Also, when he checked the heart of this mice, when he actually conducted the further analysis, he found that there was live R strain present in this particular mice. Now, this was expected, right? What was the second set of experiment? In the second set of experiment, he actually selected the live S strain. Now the S strain is the smooth strain and it has a polysaccharide code and it is virulent. That means it can cause pneumonia. Again, Griffith being an intelligent person, what did he find out? As soon as he injected the live S strain into the healthy mice, it developed infection. That means it got pneumonia. And the poor mice died. Right? And also on further analysis, he found that there was live S strain in this mice. Right? He could recover live S strain. Okay? Now what was the third set of experiment that he did? What he did was, this particular S strain is causing pneumonia, so he heat killed this particular strain. He used extreme heat and he killed the S strain. Okay, he killed the bacteria and he took this heat killed S strain and he injected it into the healthy mice. Now, of course, as I already told you, Griffith being an intelligent person, what should he expect? Since this S strain is killed, of course, your mice will not develop any pneumonia or any kind of infection and the mice will live. So did he observe that? Yes, of course. So there was no infection over here. And very luckily, our mice lived. And on further analysis, he did not find any particular live bacteria. No live bacteria was found. Now when I say no live bacteria, I'm not meaning any kind of bacteria. I'm talking about the Streptococcus strain, okay? There will be different types of bacteria in the sample, but basically he did not recover any live S strain from here, okay? So that was as expected, okay? There was no live S strain. Now in the fourth set of experiment, what did he do? Now him being very adventurous with his experiments, what he did was he took this particular heat killed S strain. Okay, the S strain is killed. It should not cause any kind of infection. The mice should remain healthy. So he took this heat killed S strain. Then he combined it with this live R strain. Okay, R strain is harmless. It does not cause any kind of infection. So he took that live R strain, mixed them together, and then he injected into a mice, a healthy mice. Again, Griffith being very intelligent, he was expecting what? Heat killed S strain does not cause infection, live R strain does not cause infection. Of course, his mice should be alive and healthy. It should not get any kind of pneumonia. So, did he actually observe that? No, the poor mice died. Actually, the mice got infected with pneumonia. That means infection. It got infected. And our poor mice actually died. So sad for that, right? It was very unexpected. And more shockingly, what did Griffith find? Instead of finding the live R strain alone in this sample, he actually found live S strain. What just happened? It just blew Griffith's mind. He was like, I had injected heat-killed S strain. How come in my mice I'm getting live S strain? How did my mice die? So that is where he actually discovered that something was transferred from this particular S strain into the live R strain, which transformed the R strain into the virulent type S. And it caused infection in this poor mice, and his mice died. One more point to be noted over here was the strain that Griffith was using, this R strain, is nothing but your type 2 R strain, okay, type 2 R strain, and the S strain that he was using was type 3 S strain. And the surprising fact over here was, even though he used type 2 R strain and heat killed type 3 S strain, in the mice, the after analysis, in the dead mice, he actually found type 3S strains. So it was not because of any kind of mutation also, because if it was kind of mutation, type 2R should have become type 2S, but no, he noticed that it has become type 3S. And that is why this discovery was so profound for him, because he found out that there is something happening, right? So this was a discovery done by Frederick Griffith. This was a set of experiments that he conducted in order to find out the transforming principle. So he named this process as transformation, and he called this something that was transferred as a transforming principle. He did not know what it was, but he called it as transforming principle. Now in the comment section, please let me know what would have happened if Griffith would have injected dead R strain along with heat killed S strain. Do you think the mice would have survived, or would it have met its end? Please let me know in the comment section. I know this question is a very basic one, but please feel free to answer it in the comment section. So that was Griffith's experiment on discovering the transforming principle. So basically, in Griffith's experiment, he noticed that something was transferred from the S strain or the virulent strain into the R strain. Something was transferred, which made the R strain virulent, and it killed the mice. So that was an amazing discovery, right? So he called this process transformation, as something was transforming the bacteria from one strain into another strain. Now this material that was transferred between the strains had to be the hereditary material, because the information was being passed, right? The information of a bacteria to be a virulent strain, that information was passed on to a non-virulent strain. So that is why this notion that whatever is transferred from this particular S strain into the R strain should be the genetic material, that particular notion came up. right? Now, what was that something? What type of substance could change the characteristics of the organism that received it? So that transforming material had yet to be identified. Now, transformation today is known as a change in genotype and phenotype due to the assimilation of external DNA or hereditary material by a cell. Now, what was the genetic material? We will try to answer in the coming up videos where we discuss about the experiment by Avery, MacLeod and McCarthy, and then later on by Hershey and Chase. So with that, I will end. And as always, I will leave you with the motivational quote. The one for today is, success is not final, failure is not fatal. It is the courage to continue that counts, by Winston Churchill. So it's as clear as that. Do not stop at your failures. It is always the courage to get up one more time that counts. Remember that destination need not be changed. You can have a destination kept in your mind, but the way or the direction that you use to reach that destination, that can be changed, right? So consider failures as those directions which did not work out, the dead ends which did not work out. Get up, move, take another direction, and keep on moving towards your destination or your goal. If you liked today's video, please do like, share and subscribe to my channel, and also please put in your valuable feedbacks in the comment section. That's all for now. Until next time, bye-bye.